So I know that we're packing a lot in, and so I do want us to move on. We're now going to move to our first panel, and the idea in setting up this panel was to try to invite leading thinkers and scholars in very different areas to talk about how various advanced technologies are affecting their work as scholars, researchers, thinkers. And so I am just thrilled to have four amazing professors and researchers with us today. We're going to go right from speaker to speaker, but I'll do just a very quick introduction. We'll start off with Professor Corbin Jones, who is professor in the biology department here at UNC and leads the integrative program for biomedical and genome sciences. He's also the faculty director of our UNC High-Throughput sequencing facility. And I think it's safe to say Corbin manages probably the largest data set on this entire campus. And we're pleased to have him with us. Professor Nikhil Kaza is professor in the Department of City and Regional Planning. He also directs the Center for Urban and Regional Studies, and his work really looks at using advanced geospatial technologies to try and make good plans for both local and global kinds of built environments. I'm sure you'll enjoy his perspectives. Third will be Professor Victoria Sabo, who's a research professor at Duke University in the Department of Art, History, and Visual Studies. And she's been thinking about and using technologies in art and media for many years. And I hope that she'll bring the humanities perspective to this discussion. And then Nancy McGovern is the director of preservation at MIT. And Nancy has been active for many years in digital curation and finding ways to advance archival practice for born digital materials. And I think you also find her experiences quite interesting. So very diverse points of view. And with that, I'm going to turn it over to Corbin.
All right, well, thanks, Gary. Oh, make sure you guys can hear me. It's really exciting to sort of take this set of questions that you have posed to us. And I assume that all of us will be taking a look at these and trying to explain from each of our, sitting or standing points, as the case may be, how we sort of see, artificial intelligence and these technologies impacting what we do. And so when I think about how I do use these technologies, there's really sort of three areas that I come into contact with them. As Gary alluded to it, genomics and the data that's being generated in genomics, the scale of data that we can produce now is so large that really we need efficient and fast ways of processing it, of learning what patterns might be within it. And just to kind of give you the scale of the data, I mean, at UNC, we can produce, a couple dozen human genomes sequenced, wholly resequenced in a day. And it now takes us actually longer to move the data from the sequencers than it does to get the data from the samples. It's really gotten to be quite critical. And from that, we're seeing AI playing several fairly critical roles. First of all, we're talking about genomics. That's one type of omics out there that's looking at genes, how much those genes are turned on, maybe factors that might be regulating those genes. But then there's the proteins that get produced from that information that actually do the work in the cell. And then those proteins get modified and changed and altered. And there's a whole field of proteomics that continues to grow here at UNC. And it's getting stronger and stronger and more in depth. And so just bringing those two data sets together, the proteomics and the genomics data sets together, is one of the areas where we really see artificial intelligence playing a critical role, trying to reflect and understand and find patterns within these data that are far too large for any human to take up. I mean, literally, we're talking petabytes of disk space. So that linkage aspect between these disparate types of omics data is very important. So sort of bringing it all together, but also equally important is sort of taking it all apart. There have been incredible advances in our ability to profile exactly what an individual, a single cell is doing at a particular moment in time. So if we grab it and do our magic on it, and we can sort of see, this is up, this is down, that's changing. As we learn these patterns about what a single cell type is, we're also learning the ability to take an amalgam of cells and say, okay, I've got this bucket, I'm going to use AI to tease all those apart and sort of understand what made up this amalgam. This bunch have got a profile, a signature, characteristics of cell type A, and this other set over here appeared to have characteristics of cell type B, and then this middle set is a fusion, maybe 60-40 of those. And that ability to sort of both integrate data using AI tools as well as disambiguate data has become very critical. And obviously, when you start thinking about any sort of large-scale biological information, you need to think about annotation and feature classification. You know, we have a project going on right now where we're sequencing a bunch of grass genomes, trying to understand better why our common cereal crops have to grow in bright sunlight, yet so many grasses grow in the shade. What are those fundamental genetic changes? This is a collaboration with Alan Jones here in biology. But from an AI perspective, what we need to do is take in these new genomes and then do feature prediction. What are the genes? What are the things regulating those genes? Is this gene similar to that gene? We've had a lot of algorithmic tools over the years to sort of do this, but with the advances of artificial intelligence, particularly machine learning, we're able to do a lot more. And sort of the other area that's really impacting us in the genomic scale has been prediction. Can we take data? And I'll give you an example of a project we're doing with Jeremy Wang in genetics and Thomas Alexander over in the Cancer Center, where we're using a very small portable genome sequencing device, about the size of a phone. And we're trying to find ways to use this to do tumor subtyping. So if you get a tumor sample, you can better target therapies and treatments based off the subtype it is. Here in the US, we have literally hundreds of $1,000 machines that allow us to do this, but in more sort of low to middle income countries, they don't have access to the same sort of tools. So what Jeremy and Thomas have been pushing is taking these very small sequencers, right, and putting them in those environments and seeing if with some clever AI techniques, we can take a very small amount of mediocre sequence data and subtype those tumors in kids. So it's particularly being applied to leukemia samples. So that's another example of how we're using this to make predictions about it and classifications about what someone's going to be. So that's from sort of the genomics perspective. If I pivot more to my personal research, it's more in population genetics. And the biggest change, and this is some work that Dan Schreider over in genetics here, who's a computational biologist, has really been on the forefront, has been using a combination of simulations and AI to take, again, a complex biological signal and try and understand the evolutionary and ecological forces shaping that genomic signature. So if we think about, I'll take a simple example, you go out and you identify a bird species that is evolving with climate change, right? And you would like to understand how much of this is migration of, let's say, more warm tolerant birds into a new region, how much of this is the current cold tolerant birds evolving to be warm tolerant birds, species, AI and machine learning has enabled new ways to sort of disambiguate the genetic signature we get out of those populations and say, aha, this seems to be pretty strong over here. This seems to be strong over here. And of course, we've got a number of other sort of larger scale collaborations looking at medical data that have been very important. I will say one particular collaboration I'm very excited to be working with is with the fine folks at the Renaissance Computing Initiative, Stan Ahalt and his team at RENC. And there we've actually decided to investigate a long-standing sort of relationship between biology and AI. You know, if you think back, genetic algorithms, neural networks, all these sorts of things are really analogies from biology that have been brought to sort of shape the techniques that we're using in artificial intelligence. And so something that Stan and a team of folks at RENC, as well as some of us biologists, have been trying to do is see if we can use the biological processes that give rise to epiphenomenon in interesting ways to guide the development of new AI techniques. And so this has been a fun little project. All right, but jumping ahead, the major shift I would say we've seen, you know, one of the next questions is how has our work changed over time and which of these changes are due to AI and other similar techniques? The biggest change has been a movement more to these black box AI models and maybe a movement away from the classical sort of, you know, analytical or mathematical modeling that we used to do a lot of. Looking forward, I actually think the future for AI is going to involve sort of a fusion of these two things. And we can maybe talk about that a little bit later on. In terms of the daily practice of our field, I think we're seeing lots of new algorithms. We're seeing lots of large, giant data sets that people are applying more and more sophisticated tools to. So on one hand, I see AI making a huge impact. On the other, because you still need to have the rubber meet the road, you need this ability to actually go out and prove or test the hypothesis you need. We're now limited often by what are the experiments we can do to test the ideas that are coming out of AI. I'll give you an example of that in some collaborative work with Victor Garcia, looking at the impact of SARS-CoV-2 in the infection of the lung. You know, we've been able to use some of these classification techniques to make predictions about certain cell types expanding or contracting over time inside the infected lung. And some of these we can go out and prove just by taking sections of lung tissue and using, you know, chemical reagents and other things to identify those key cell types. And then you just count them. Are there more? Are there less? But that presumes that we have those reagents, that we have the thing we need to stain the type. And while we've been able to do this for one cell type, another one's been very frustrating because we haven't been able to get that critical resource. So again, AI is giving us lots of hypothesis generation. It's assisting in sort of linking and classifying knowledge. But that fundamental fact that as a biologist, we still need to do experiments is limiting what's going on. So one of the next questions was, what am I excited for in the future? Well, as I intimated before, the RENC project, where we're really trying to go back to trying to shape new AI with biology and have new AI apply and bring out new patterns in biology, that has me really excited. I hope we can get that to go. I'm, of course, a bit skeptical about how much AI will fundamentally change for those experimental reasons before how we do the practice of biology, because we still need to make that final linkage. So I think this is critical. Okay, and so the last thing, Gary, you asked, or you sort of suggested we discuss, is what are my suggestions for educating future generations of people who work in knowledge-intensive fields? Well, first of all, I think as biologists, we have to embrace the fact that this is so much more data-driven. For years, you know, biology was sort of seen as a refuge for STEM-inclined students who didn't want to do too much math, right? That's gone. Frankly, that's over. Biology has created a quantitative biology track. I think we're going to only see that grow. But in terms of practical things we can do as a larger AI community, I think micro-credentialing, boot camps, workshops, hackathons, these are the ways that we engage with the students out there to get them those little pieces of knowledge that they can build on. I mean, one of the problems is getting a broad community of thinkers engaged in AI. So having good ways for them to just try it out. Maybe we can get those who weren't naturally or initially interested in it hooked onto it. And as much as we want to teach people these techniques and these approaches to doing AI, I think it's extremely critical that we teach them how to think critically about what it's doing. We all know the examples of invading pixels and these biases that come up, let's say, in image processing and analysis. How do we teach students to see those ahead of time, to predict those, to understand why the algorithms are doing that and alter that? That is a hard task because it's not just teaching them better ways to code, it's teaching them how to think deeply about the theory and the ideas behind AI. And the last thing I would say, we got to give kids a chance to work with real world data. We got to let them get their hands on it so that they can get involved at an early stage and an early level. I have found that the opportunities for students to get involved in research have contracted, I feel, in recent years. We need to be able to support students who don't necessarily have these tools in their hands today through those workshops, like the hackathons, like the Pearl Hacks here at UNC, which was a great operation, so that a broad range of students from many different backgrounds can get pulled into the power and utility in a critical way of AI. And so that's what I have to say.